Hi everyone, my name is Romain Lopez, and I'm a postdoctoral fellow at Genentech and Stanford, and I will start my lab next year in September at New York University at the Current Institute in Computer Science and at the Department of Biology. Today I will talk about optimal transport and perturbation screens. So I will not talk about sequence models. I hope that's all right. All right. So this was a team effort that was led by Jiayong during her internship at Genentech. And this was also a collaborative effort with Charlotte, Luca, Pinello, and Jiayong and Luca both at Harvard Medical School, and also with Aviv at Genentech. This work we were pretty humbled to receive the Base Paper Award at the ICML workshop in AI for Science earlier this year. So I will start by motivating why optimal transport can be important in this specific context, and I will talk about the specific problem I'm interested in here. Then it will motivate the method, and I'll go through some of the algorithmic advances here. Talk about the baseline that's pretty largely and broadly applicable and that we could try here. And then we'll see on some multimodal data how this performs. So let's get started. So high-content CRISPR screening have been very valuable and has really transformed how we think about functional genomics. When I talk about high-content CRISPR screens, I usually think about cell lines or primary cell that express CRISPR. And we transduce, we deliver guide RNA to the cells to be able to edit the genome and basically have those knock-in, knock-out, or knock-down some genes inside of the cells. We obtain, it's called a pool screen, so we obtain a bunch of cells with different perturbation inside of them. And then I write here complex phenotype, meaning there are different ways of then measure some biological insight in those cells, and I will go through different of those measurements, and we'll think about them as modalities. So this kind of framework is very powerful and has been applied for understanding genetic interactions, understanding mechanisms of cancer resistance, immune cell function, as well as cellular differentiation. It's important to recognize that so there are different ways, again, of profiling this pool of perturbed cells, and each modality has its own strengths and limitations. I will talk about two specific modalities. One that's very popular is called Perturb-seq, in which we will basically perform single-cell RNA sequencing on this pool of cells, and we can also retain the guide information, so we know what we perturbed and we know what is the outcome in terms of gene expression. We can say that the readout is unbiased because we observe all the genes in the transcriptome. It's relatively safe to say that it's interpretable in terms of outcome, because if we put a few genes together based on pathways, we can have a broad sense of which pathways are up and down regulated after those perturbations. And it's also very safe to say that it's relatively expensive and difficult to scale. So there is an alternate type of modality, which is based on microscopy imaging. And this assay called cell painting consists in taking those cells, dyeing with chemical dyes the organelles, or some organelles with some broad set of markers, and putting them under the microscope to see what the morphology of the cell looks like. And we can somehow featureize it. And it's interesting to think about it as also an unbiased readout, because even if you have enough of those organelles, even if they're not very specific in terms of pathway, they still give you a broad sense of how the cells are changed after perturbations. It's very difficult to interpret them. For example, if you think about immune cells and then you have those broad markers, you will not be able to read off directly what you're interested in, but it might still contain that information. So let's leave it at it's not super interpretable, but it's very scalable in terms of number of perturbations we can profile, and potentially we can do genome-wide screens in this kind of setting. It's not very difficult. So this motivates the idea of can we get put this data together to try to get in silico Perturb-seq, for example, for many more perturbations by diminishing the cost of those experiments. So as a mental thought, I guess, that would work as follows. For many perturbations, we can profile single-cell RNA sequencing as well as imaging. We learn a model that will help us translate from one modality to another. So here, for the exercise, I will pick predicting gene expression from imaging. And once we have trained that model on the data that we have, we can basically pick the other perturbations for which we have only one modality and try to predict and fill this in. And we can call that in silico perturb-seq from the imaging data. So how the most important and critical step here is how do we build this modality translation model? And let's try to, again, go through a very cartoon type of example. What's happening here is basically we measure for several cells some features or some cloud points. And we have two modalities, so two different spaces that are broadly incomparable. They live in different scales, etc. Each point is a cell, and we have colors that correspond to somehow labels or perturbation. And so I can take a particular cell, I can have some kind of model or a neural net that helps me pass, translate, and go through the other space. I arrive somewhere, I don't really know where I am, and the reason is I don't really have paired measurements. So these methods are destructive in a sense, meaning that I can never recover the exact cell that is in the matching modality. So let's say I arrived in another cell, and that measurement is called yj, starting from cell I. And I can't really use that for a supervision signal, for example. But maybe if I have an oracle or somebody that's very nice to me telling me, hey, I think those cells are actually related, or maybe those cells are completely unrelated, and I have a matching matrix, T, then I can somehow still, as a starting point, write an objective function, which is a weighted nonlinear least square type of problem. So I try to predict one modality based on the other and kind of weighting those pairs of observation based on how relevant they are. And I'll call this T matching matrix. I want T to be doubly stochastic, meaning that it kind of is a probability distribution for every cell. I associate a probability distribution over the other cells on the other modality. So this is kind of the setting. Now, everybody will look at me and ask, how do we get this matching matrix? So I don't know yet, but what we can first do is we can go through the exercise of thinking, what if I have paired data? So if I have paired data, I will pick the identity matrix because I know how to pair the cells. And then I know that my model F will be basically learn with supervised learning, and it's kind of the best thing we can do, and there is not much interesting things to be done here. Now, if it's not the case, maybe we have some labels, and we can say, when I pair my observations like this across modalities, I want to make sure that for every cell, I try to predict the sequencing based on the imaging of only the cells in the other modality, but that have the same label. And when I do these kind of things, I'm kind of doing some shrinkage. So implicitly, I will not be able to distinguish at the single cell resolution anymore, but I will be able to perform some kind of pseudo-bulking. So I will lose resolution, but at least I have some information. And the point is, can we try to do better than that? Okay, so this is a minimal kind of thing we can do. And so we need to estimate a matching matrix for doing this. And optimal transport in general is a really nice framework for learning matching, except that in general, optimal transport thinks about, or it's in it's the easier formulation, usually cares about matching within the same space. I'm going to first go through this for a few minutes. We have two probability distributions that live in the same space, mu and nu, and we want to know how to fit a coupling matrix, a pi, or a transport, or a matching, that will tell me how to move mass from one part of the space to another based on a certain cost. So if I don't care about the cost, the problem is defined as follows. I can pick any admissible matching matrix that basically matches, that corresponds to the right marginal distributions. I want to make sure I move all the mass from one place to the other place. But now, out of all these possible matrices, coupling matrices, I want to make sure that I find the one that minimizes the cost. So I can write it as an optimization problem, and that's usually how optimal transport is formulated. We obtain this optimal plan or optimal matching, and the value of the cost, the minimal cost, will be called the Wasserstein distance. And usually we don't quite run it like this. We will solve a regularized problem with entropy, and that allows for going into the dual space, making it strongly convex as a problem, and having much faster inference procedure. So this has been applied very widely to single-cell data and has been presented in this kind of venue many, many times. So I'll go through it quickly. This is not useful, again, because we cannot use that when it's across different spaces. So people have worked on the Gromov-Wasserstein optimal transport, which allows for matching across comparable spaces. The way that this works is basically instead of having a cost function across two spaces that we do not know how to construct, we build a cost of cost functions. So basically, we look at the distance matrices within each space, and we try to minimize displacement of the cost matrices. And intuitively, what these kind of algorithms are doing, they're doing shape matching. So they're trying to make sure that using the singularity in the shape, somehow we can kind of learn a decent matching. And so this has been applied also to single-cell data in the context of matching RNA-seq to ATAC-seq and other modalities like this when you have completely unpaired measurements, which looks like what we're trying to do here. But not quite, because we have some extra information, and that's the purpose of this presentation. So we're trying to get to the point where we can actually include information from the labels. And so that's what we dubbed label-constrained optimal transport. And the key idea is, instead of just doing naive shape matching, can we use the perturbation labels as anchors across multiple modalities? And in the context of optimal transport, if we look at the mathematical framework, maybe we can somehow just constrain the optimization problem to match samples that are only within the same label. So it doesn't sound too hard written like this. And that's our contribution. So we extended two frameworks for matching across modalities, Gromov-Wasserstein OT and co-optimal transport, for doing this kind of label-constrained method. So what we're interested in FINE is actually to understand, if we have a better matching, can we predict better across modalities, right? So I'll get to that throughout in a couple of minutes. So to finish, for this part of the talk, it's important to think about what's our setup. Our setup is, we have, again, two modalities. First, we will run this label-constrained optimal transport procedure that I haven't yet described. And then we will use that to learn a predictor and do the modality translation. All right. So now we can look into the specifics of how this label-constrained optimal transport can work. And so first, you might wonder, can I just split my data sets per label and then run number of label times a Gromov-Wasserstein optimal transport? And do I get the same, and isn't it what you want to do? The key answer is no. So it's, the optimal transport problem could break up like this, but Gromov-Wasserstein is a bit different, and this slide is meant to explain why. So if you only consider per label or per perturbation Gromov-Wasserstein, I'm showing you here two cloud of points. And the distances or the arrows that you can see are kind of like what you would do for what you would compare across modalities for the Gromov-Wasserstein matching. And here it looks like the cell I have on the corner is kind of matching really nicely the distances of other cells picked in the other modality. So I would be very tempted to somehow put a high matching probability between those pairs of cells. But if I had other labels, maybe perturbations that affect another pathway or downregulate the same pathway, I'd be able to kind of figure out that actually that cell is very close to cells from another label in one modality, but very far from the same cells on the other modality. And in that case, if I just split the data set, I kind of lose the information. All right, I hope that makes it clear. If you look at the math, it's also clear, but I think that the drawing makes it nice. So now let's try to get into how the algorithm works and how we can quickly change it for solving that particular problem. So when you think about entropic Gromov-Wasserstein optimal transport, Gabriel Perret and other authors in their paper explained that you can solve Gromov-Wasserstein OT under some assumptions about the cost function that we will assume are fine here. You can break down this problem into a sequence of optimal transport problem across the same space by kind of changing the cost function. So you have a iterative dependent cost function, and you solve like this a sequence of OT problems. And this is equivalent to some kind of projected gradient descent. So you can write the algorithm like this, basically. The algorithm would look like I calculate a cost function or a cost matrix. I solve optimal transport with this sinkhorn iteration. and then I go back, calculate the cost again, and perform synchrone iteration and iterate until I converge. And I know I solved the Gromov-Wasserstein optimal transport. So this is for the unlabeled setting. And our contribution is just to say, all right, because somehow what we want to do is make sure that I cannot match things when the labels are different. I just need to do an extra projection. My problem is convex, so it's nice. My constraints are convex. I take the intersection of things convex, it's still convex. Things work out. And somehow, now I can just need to think about how do I do this extra projection. And it turns out we can still solve the OT problem with the same synchrone iterations. They're just slightly different. So you can see here we need to kind of like multiply by this matrix of compatibility that tells you if you're in the same label or not. That's doing the projection for us. And the cost function is also a little bit different and has a better runtime, but I'll leave it at that for this short presentation. We were able to implement, so Jiayong implemented this in the OTT JAX framework. It was really nice that we were able to work with Charlotte Bune because she implemented part of that package, and she's been a postdoc with us at Genentech and now started her own group at EPFL. We get this type of methods to work. So I'll present a natural baseline that everybody would try to apply to this kind of problem with a variational autoencoder. So, and we've seen, I guess, some of those models in different contexts maybe today, but you can use those autoencoders with different heads. So basically, to resolve this shape matching problem. So here I'm going to propose one way, but there could be many. You have two autoencoders that are trained at the same time, one for reconstructing one modality, one for reconstructing the other. And then you learn them jointly and force the bottleneck layers, or the latent variables, I would rather say that, but the language is fine. You use those bottleneck layers and you kind of force them to be undistinguishable in distribution from each other. So you could use an MMD, or here you can use an adversarial classifier. And if you train this jointly and you force things so that you cannot distinguish one modality from the other in the latent space, then you basically solve this problem. And it's been applied in recent papers working on also imaging and sequencing in a different context. So this is a nice baseline to try, but it's not label aware. The way to make it label aware would be to have a different adversarial classifier per label, which adds a lot of parameters. So to kind of avoid doing that, we just added the label of the one hot encoding of each perturbation label into the classifier. And it's a crude approximation, but it's a baseline that we're trying to apply here to get a sense of how much better things get with adding labels. Right, so now I'm going to discuss our application to the real data. The bad news is we don't have real data of images and sequencing. Also, either way, it wouldn't be paired, so it would be tough to assess a matching algorithm. So I'm going to use a surrogate type of data. So this is data produced in the context of a collaboration with Professor Kelvin Chen at Osaka University. This is a chemical screen data set where we take T cells and activate them with anti-CD3, anti-CD28 antibodies, but in the context of small molecules. And the assay works like this. So we take those CD4 T cells, put different drugs, activate them, and measure protein and RNA sequencing. We filtered a few drugs that have the highest effect to test the method. We also have dosage information that's kind of helpful. It will come in later to visualize and make sure that the algorithm is behaving properly. And we're trying to predict gene expression from proteins. So I will argue that it's quite different from the original setting about imaging and sequencing, and it's much simpler, but it's a first step into evaluating this kind of method. So if you look onto, I guess, some kind of UMAP space, I'm plotting here different drugs and the gene expression space and protein space, and you can see that when you increase the dosage, things are moving along in latent space, which is kind of giving you the idea that the drugs are having an effect on the cells. I'll leave it at that for visualization. So the experiments we wanted to perform was, first we want to run the OT-based methods. Those methods are trained on the 50 PC latent space for both gene expression and proteins. We run optimal transport, Gromov-Wasserstein optimal transport, and this other framework I haven't discussed called co-optimal transport. For all of them, we run the per label, no label, and label constrained. And then we train a multi-layer perceptron to predict the gene expression from the proteins using the same formula I've discussed. And then we also compare to the adversarial VAE. The architecture was based on multi-VI. And there are some hyperparameters you need to optimize. So for the optimal transport, usually the entropic regularizer is critical. So and for the adversarial VAE, you need to pick the adversarial weighting of the penalty in the elbow. And so what we did was folded 5-fold nested cross-validation, in which we picked the best result by splitting and average the results across all the folds on the test data, of course. And so to evaluate, we evaluate the matching because we have paired measurements, and we look at the fraction of samples closer than the true match. We also look at the dosage matching. So the dosage was not given to the algorithm. The labels were only the drugs. And we look at whether the model can figure out that cells from the same dosage should be matched to each other more closely than otherwise. And we look at how well we can translate those modalities, so based on fold change of gene expression and correlation also across cells, and the mean square error. So the results look as follow. First, we have the baseline, where we have the one-to-one mapping, the per dosage mapping, and the uniform per label, which is what the algorithm only sees the labels. We can see that the coarser information as expected, and the worse the results get. We can also see that the entropic optimal transport, because it doesn't operate in incomparable spaces, doesn't perform well at all. But we see that the variants that we propose with the labeled data actually have decent performance and are the best contenders in this study, both for Gromov-Wasserstein and co-OT. And the domain adversarial VAE performs decent matching, but has really poor performance for prediction, which means likely there are some. These methods are really tough to make work. There are a lot of not just maybe the lambda was not the only hyperparameter we should have tuned, and also the number of epochs you train when you train the adversarial classifier, etc. So it's but this is the first try. So as a conclusion, I would like to say that those labels constrained version of OT are interesting, and they improve prediction results for this cross-modal perturbation data translation project. What's important to think about is what is the input space for those models. So in general, you really need to, because we're doing shape matching, you really need to have the latent space in the right form, meaning that if somehow you have technical noise on the imaging and technical noise on the sequencing, those induce completely different shapes. When you're going to apply those methods, things are going to fail pretty hard. And then the last thing I want to say is we're looking into amortization and neural optimal transport for increasing the scalability of those methods as well. And we're excited to apply them on this problem with real data of imaging and sequencing. And thank you very much.
Hey, thanks for the nice talk. I'm curious, going back to your original problem of mapping Perturb-seq to self-painting, how do you compare your general class of methods to methods where you just featurize by aggregating over cells? Is there obvious evidence that sort of you need to model that individual cell level?
Yeah, so that's a good. I think that's a good point. I think if you start having enough perturbations and many contexts, maybe at some point you start picking on variation that will even be inside of the bulk variation of the control cells, meaning like cell cycle and all this stuff. So it's possible that with enough data and enough perturbation, you start getting into that. But I think the way I think about the single cell variation is that a lot of it could also be like confounding factor that could lead your algorithm astray when you're trying to make predictions. So the images across the cell cycle look very different, for example. But the genetic is genomics data as well. So if you can kind of condition on this and pick it up from an optimal transport like model, you might reduce your variance and have better results in maybe in a lower regime of less perturbations. So that's how I'm thinking about it. Yeah.
Hi, great talk. How would you suggest somebody scale it for multiple modalities, let's say like 4 or 5? Would it be just pairwise for each, or is there a more optimized method?
That's a good point. I didn't really think about this. So if you start having, so yeah, you could either do all the pairs. The way that would make more sense is to, once you start having many more modalities, is to think about some kind of common space, I guess. But in the context of optimal transport, I don't know exactly what it would mean. You need to define some kind of, that wouldn't fit in that exact framework. Maybe with autoencoders, I think people have already done this kind of things.
Okay, thank you so much.
Thank you.